The Lyginopteridales were the archetypal pteridosperms. They were the first plant fossils to be described as pteridosperms and, thus, the group on which the concept of pteridosperms was first developed. They are the stratigraphically oldest known pteridosperms, occurring first in Late Devonian strata, and they have the most primitive features, most notably in the structure of their ovules. They probably evolved from a group of Late Devonian progymnosperms known as the Aneurophytales, which had large, compound frond-like leaves. The Lyginopteridales became the most abundant group of pteridosperms during Mississippian times, and included both trees and smaller plants. During early and most of Middle Pennsylvanian times, the Medullosales took over as the more important of the larger pteridosperms, but the Lyginopteridales continued to flourish as climbing, lianescent, and scrambling plants. However, later in Middle Pennsylvanian times, the Lyginopteridales went into serious decline, probably being outcompeted by the Callistophytales that occupied similar ecological niches but had more sophisticated reproductive strategies. A few species continued into Late Pennsylvanian times, and in China persisted into Early Permian, Asselian times, but then became extinct. Most evidence of the Lyginopteridales suggests that they grew in tropical latitudes of the time, in North America, Europe, and China. As the Lyginopteridales are the earliest known gymnosperms, and the development of ovules was one of the key innovations that enabled seed plants eventually to dominate land vegetation, the evolution of Lyginopteridalian ovules has attracted considerable interest from paleobotanists. The most important work on these early ovules was by the British paleobotanist Albert Long, based mainly on early Mississippian, anatomically preserved fossils from Scotland, UK. Some of the earliest ovules, example Gymnosperma, consisted of a nucellus, the equivalent of the sporangium wall, surrounded by a sheath of slender axes known as a pre-integument. It is widely thought that this arrangement was derived from an ancestral condition where there was a cluster of sporangium-bearing axes, but where only one eventually retained its megasporangium, the others forming the surrounding pre-integument. Progressively, this sheath of axes became fused to form a continuous integument that surrounded and protected the nucellus, such as in Eurostoma and then Stamnostoma. These earliest ovules had the apical part of the nucellus exposed, from which there was a projection known as a logonostome, sometimes also called a salpinx, that facilitated capture of the pollen and directed it down to the pollen chamber above the megagametophyte. In later Pennsylvanian age ovules such as Lyginostoma, the nucellus became almost entirely encased by and fused to the integument, leaving just the small distal opening in the integument known as the micropyle through which pollen passed. Nevertheless, most Lyginopteridalian ovules retained a logonostome, despite its function in pollen capture having been replaced by the micropyle. As with all seed plants, the Lyginopteridalian ovules had just one functional seed megaspore within the nucellus. In some, however, 3 other, aborted megaspores were still present that together with the functional megaspore represented the remains of what would have been the tetrad of megaspores in the ancestral pre-seed plants. Most if not all Lyginopteridalian ovules were born in an outer protective sheath of tissue known as a cupule. There are some Lyginopteridalian ovules that have been reported without a cupule, but these may simply have been shed from the cupule before being fossilized. In Late Devonian and early Mississippian species, the cupule contained several ovules, but by Pennsylvanian times there was normally just a single ovule per cupule. A number of cases have been found of cupules occurring in clusters at the ends of branching axes. Many paleobotanists now interpret these clusters of cupulate organs as fertile fronds, in which the cupulate tissue was derived from the laminate part of the frond that surrounded the ovule and thereby provided added protection for it. Most Lyginopteridalian ovules were radiospermic. The only notable exceptions were a distinctive group of Mississippian-age Lyginopteridaliums that had platyspermic ovules and are referred to the fossil family the Eospermaceae. The Lyginopteridales produced small trilete pre-pollen that superficially resemble the spores of non-seed plants but with a fundamentally gymnosperm-like wall structure. The pre-pollen was produced by sporangia that formed regular clusters, synangia. The stratigraphically older Lyginopteridalians had trusses of synangia borne on slender axes, which were attached to vegetative fronds. These are referred to the fossil genera telangium if they are anatomically preserved or telangiopsis if they occur as impressions. The more primitive forms of telangium had sporangial walls that were essentially uniform in thickness. In stratigraphically younger telangium species, however, the side of each sporangium that formed the outer surface of the synangium tended to be thicker than the inwards-facing wall, suggesting that the sporangia split to release the pre-pollen along this inner wall. In Pennsylvanian age Lyginopteridalian synangia, The sporangia were usually attached to a pad of tissue that was probably homologous to a pinnule on the vegetative fronds. For instance, Crossotheca synangia had an elongate, epaulette-shaped pads that were arranged in a pinnate pattern in a frond that was wholly or partially fertile. The Pharaxotheca synangium, in contrast, had a much less elongate, almost radial pad. The Mississippian-aged Lyginopteridales tended to have a simple protostele usually surrounded by secondary wood, but in later forms there was a eustele with a central core of pith or mixed pith. In most cases the amount of secondary wood was limited, suggesting they were stems of scrambling or climbing plants, but some Mississippian-aged forms, example pitties, had substantial secondary wood and was probably the trunk of a large tree. The stele is surrounded by a zone of cortex, which in many genera contains bands of fibrous tissue. This fibrous tissue often results in distinctive markings on the surface of the stems even when preserved as impressions and can help with their generic identification. Lyginopteris, for instance, shows a mesh-shaped patterning on the surface of the stems, whereas Heterangium has mainly transverse bars. As with most pteridosperms, fragments of the foliage are the most commonly found fossilized remains of the Lyginopteridales. When found complete, the fronds always seem to have a main rachis that dichotomizes in the lower, proximal, part. In some cases, the two branches each underwent a second dichotomy, resulting in what is termed a quadripartite frond, but in others there is just the main proximal dichotomy, resulting in a bipartite frond. The branches produced by these dichotomies then undergo further divisions in a pinnate manner similar to that seen in fern fronds. The ultimate segments, pinnules, of the fronds are mostly lobate or digitate. Various fossil genera are recognized for these fronds, distinguished on whether the frond was bipartite or quadripartite, whether there were pinnae attached to the main rachis below the main dichotomy, the general shape of the pinnules, and the surface markings on the rachises and stem reflecting the sclerotic tissue in the cortex. There is no consensus on the division of the Lyginopteridales into families, either in terms of whole organisms or as fossil families of particular plant organs. The most recent scheme, by Anderson et al., 2007, recognized 5 families based on ovulate structures. Some authors also recognized the Myriopteridaceae for the distinctive group of Pennsylvanian-aged lianescent plants with Myriopterous fronds, although details of their reproductive structures are unknown. References